guys, welcome to BIOGURU. Now we will discuss about the transcription. So transcription क्या था? The process of copying genetic information from one strand of DNA into RNA is termed as transcription. DNA की 2 strands में से एक strand से RNA formation होगा और इस process को बोलते हैं transcription. Here, the principle of complementarity governs the process of transcription, except that the adenosine now base pairs with the uracil instead of thymine. Because DNA contains A, T, G, and C, and RNA contains A, U, G, and C. So, however, unlike in the process of replication, in which once sets in, the total DNA of an organism gets duplicated. In transcription, only a segment of DNA and only one of the strands is copied into RNA. DNA में complete DNA content replicate हुआ, replica बनी, copy बनी, but in transcription हमारे DNA का एक छोटा सा segment, जो छोटा सा segment जिसे आप जीन भी कहते हो, वो RNA फॉर्म करेगा and दोनों strands में से केवल एक ही strand से RNA formation होता है. Firstly, if both strands act as template, अगर दोनों ही strand से RNA फॉर्म हो जाए, दोनों ही strand template की तरह act करें, they would code for RNA molecules with different sequences. अब दो अलग-अलग sequences बनेंगे because they are complementary, not exact copies. And in turn, if they code for proteins, the sequence of amino acids in the proteins would be different. तो अलग proteins बनेंगे. Hence, one segment of DNA would be coding for 2 different proteins, and this would complicate the genetic information transfer machinery. तो अगर एक जीन के लिए 2 different proteins बनेंगे, तो जो अब translation या transcription machinery है वो काफी ज़्यादा complicate हो जाएगी. Then why both the strands are not copied during transcription? तो अभी एक तो आंसर आपने पढ़ा. Secondly, the 2 strands of RNA, अगर दोनों स्ट्रैंड्स कॉम्प्लीमेंट्री हैं तो आपस में कॉम्प्लीमेंटेशन होगा एंड डबल स्ट्रैंडेड आरएनए फॉर्म कर लेंगे, और अगर डबल स्ट्रैंडेड आरएनए फॉर्म हो गया देन ट्रांसलेशन नहीं हो पाएगा. सो द एक्सरसाइज ऑफ ट्रांसक्रिप्शन वुड बिकम अ फ्यूटाइल वन. सो ट्रांसक्रिप्शन यूनिट. अ ट्रांसक्रिप्शन यूनिट इन डीएनए is defined primarily by the 3 regions: promoter, structural gene, and terminator sequence. There is a convention in defining the 2 strands of the DNA in the structural gene of a transcription unit. So, since the 2 strands have opposite polarity, 3' to 5' and 5' to 3', and the DNA-dependent RNA polymerase, transcriptase, also catalyzes the polymerization in only one direction, that is 5' to 3'. तो ट्रांसक्रिप्शन का डायरेक्शन भी क्या रहेगा? 5' to 3'. The strand that has the polarity 3' to 5'. तो दोनों स्ट्रैंड में से जिसकी पोलैरिटी 3' to 5' है वो टेम्पलेट की तरह एक्ट करेगी and also referred to as the template strand. Then the other strand which has the polarity 5' to 3' and the sequence same as RNA, but difference कहाँ आएगा? थाइमीन के प्लेस पर यूरेसिल is displaced during transcription. अब देखिए, जिस स्ट्रैंड ने किसी भी तरह का कोई प्रोटीन कोड नहीं करा, आरएनए फॉर्म नहीं किया, उसको हम कोडिंग स्ट्रैंड कहते हैं. Strangely, this strand is referred to as the coding strand, which doesn't code for anything. All points while defining a transcription unit are made with reference to the coding strand. तो कोडिंग स्ट्रैंड के बेसिस पर ही आप प्रमोटर एंड टर्मिनेटर सीक्वेंस की बात करें. टू एक्सप्लेन द पॉइंट, a hypothetical sequence from a transcription unit is represented below: ATGC, 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 TACG, 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 3' to 5' and 5' to 3'. So 3' to 5' is tablet strand and 5' to 3' is coding strand. The promoter and terminator flank the structural gene in a transcription unit. तो एक तरफ promoter, दूसरी तरफ terminator sequence, और बीच में structural gene or you can say functional gene. The promoter is said to be located towards 5' upstream of the structural gene. Then it is a DNA sequence that provides the binding site for RNA polymerase. And it is the presence of a promoter in a transcription unit that also defines the template and coding strand. जो प्रमोटर है, प्रमोटर बहुत जरूरी है. प्रमोटर का वर्क क्या है? प्रमोटर पर ही आरएनए पॉलीमरेज आकर बाइंड करता है. By switching its position with that of terminator, the definition of coding and template strands can be reversed. The terminator is located towards 3 prime and okay, तो यहाँ पे ये reserved लिखा हुआ है, it is actually reversed. ठीक है, तो यहाँ हमें reversed पढ़ना है, it is reversed. So now the terminator is located towards 3 prime and downstream of the coding strand, and it usually defines the end of the process of transcription. There are additional regulatory sequences that may be present further upstream or downstream of the promoter. कुछ sequences just like inhibitor, enhancer, repressor, insulator, कुछ ऐसे sequences होते हैं जो promoter के work को, transcription को control करेंगे, regulate करेंगे. So now देखिए ये हमारे पास एक यूनिट है प्रमोटर, बीच में स्ट्रक्चरल जीन है, टेम्पलेट स्ट्रैंड, कोडिंग स्ट्रैंड एंड टर्मिनेटर सीक्वेंस. नाउ वी विल डिस्कस अबाउट द प्रोसेस ऑफ द ट्रांसक्रिप्शन. ओके, सो यू कैन सी इट इज अ बैक्टीरियल सेल. And bacterial cell have cell wall, nuclear region, ribosome, etc. न्यूक्लियस के अंदर डीएनए है, प्रमोटर -35 सीक्वेंस जहां पे आपका आरएनए पॉलीमरेज इजीली बाइंड करता है. आरएनए पॉलीमरेज विद सिग्मा फैक्टर, इट इज कॉल्ड होलो एंजाइम. इट विल बाइंड विद द प्रमोटर and they will form closed binary complex. Then RNA polymerase open करेगा, it is open binary complex, transcription bubble बनेगा. प्रमोटर क्लियरेंस का मतलब क्या है? जब प्रमोटर को छोड़कर आरएनए पॉलीमरेज आगे बढ़ जाता है, इट इज कॉल्ड प्रमोटर क्लियरेंस. अब टेम्पलेट स्ट्रैंड से कॉम्प्लीमेंटारिटी के बेसिस पर न्यूक्लियोटाइड्स आके अरेंज होंगे and they will form RNA strand, and it is called elongation. After elongation, RNA polymerase will reach towards the terminator sequence. Even prokaryotes में translation साथ में simultaneously चलता रहता है. And terminator sequence पर पहुंचने के बाद एक प्रोटीन आएगा, it is called rho protein, and rho protein is responsible for the termination. So rho dependent termination. Rho protein आके बाइंड करेगा ongoing RNA strand पर, and it will terminate the process of transcription. So now some MCQs. Process of copying genetic information from one strand of DNA to RNA is termed as replication, transcription, translation, or reverse transcription. Yes, it is transcription. That's good. RNA strand is synthesized from coding strand, lagging strand, template strand, all of the above. It is template strand. That's great. So transcription unit and the gene type of RNA and the process of transcription. देखिए transcription process वैसे आपने वीडियो में समझा भी. So a gene is defined as the functional unit of inheritance. Though there is no ambiguity that genes are located on the DNA, because the functional segment of DNA is called gene. It is difficult to literally define a gene in terms of DNA sequence. क्यों? Because जो DNA sequence, जो functional DNA sequence है, वो भी completely protein form नहीं कर सकता. Because यहाँ पर भी कुछ जो RNA form होगा, इसमें भी कुछ functional and non-functional part होते हैं. These are called introns and exons. The DNA sequence coding for tRNA or RNA molecule also define a gene. So, the structural gene in a transcription unit can be said to be monocistronic. mostly in eukaryotes, or polycistronic, mostly in bacteria or prokaryotes. A cistron is a segment of DNA coding for a polypeptide. Monocistronic, one gene, one transcript, monocistronic. Many gene, one transcript, polycistronic RNA. In eukaryotes, the monocistronic structural genes have interrupted coding sequences. The genes in eukaryotes are split. So, exons are said to be those sequences that appear in matured or processed RNA. The coding sequences or express sequences are defined as exons. So, exons are the functional part of RNA and introns are non-functional actually. The exons are interrupted by the introns. Exon, intron, exon, intron. So, introns or intervening sequences do not appear in matured or processed RNA. That's why RNA requires processing. The split gene arrangement further complicates the definition of a gene in terms of a DNA segment. So, inheritance of a character is also affected by the promoter and regulatory sequences of a structural gene. Hence, sometimes the regulatory sequences are loosely defined as regulatory genes, even though these sequences do not code for any RNA or protein. जैसे आपको बताया हमने कुछ enhancer, then inhibitor, repressor, insulator, कई ऐसे regulatory terms use किए जाते हैं. Types of RNA and the process of transcription. In bacteria, there are three major types of RNA: mRNA, tRNA, and rRNA. Messenger RNA, transfer RNA, and ribosomal RNA. All three RNAs are needed to synthesize a protein in a cell. And तीनों टाइप के RNA के लिए एक ही RNA polymerase enough है. So mRNA provides the template and tRNA reads the genetic code and brings specific amino acids, and rRNA play a structural and catalytic role during translation. So there is a single DNA dependent RNA polymerase that catalyzes transcription of all types of RNA in bacteria. RNA polymerase binds to the promoter and initiates transcription. It is called initiation because it have three processes: initiation, elongation, and termination. It uses nucleoside triphosphates as substrate and polymerases in a template dependent fashion following the rule of complementarity. So it somehow also facilitates opening of the helix and continuous elongation. अब आरएनए पॉलीमरेज जब डीएनए पर आके बाइंड करेगा, उसको ओपन करेगा एंड एलोंगेशन शुरू होगा. Only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA falls off. अभी सब कुछ अपने वीडियो में देखा. So also the RNA polymerase. इसके बाद RNA polymerase भी अलग हो जाएगा और जो nascent RNA है वो भी सेपरेट हो जाएगा. This results in termination of transcription. वैसे तो दो टाइप के टर्मिनेशन होते हैं, रो डिपेंडेंट एंड रो इंडिपेंडेंट, बट हमें अभी रो डिपेंडेंट ही डिस्कस करना है. The RNA polymerase is only capable of catalyzing the process of elongation. It associates transiently with initiation factor, that is called sigma factor, and termination factor, rho factor, to initiate and terminate the transcription respectively. RNA polymerase sigma factor के साथ है, initiation complex है, and RNA polymerase rho factor के साथ है, then it is termination complex. So association with these factors alter the specificity of the RNA polymerase to either initiate or terminate. In bacteria, the mRNA doesn't require any processing to become active, and also since transcription and translation take place in the same compartment because there is no separation of cytosol and nucleus. Many times the translation can begin much before the mRNA is fully transcribed. कि जो ट्रांसक्रिप्शन है वो कंप्लीट नहीं होता, उससे पहले ट्रांसलेशन स्टार्ट हो जाता है. Consequently, in the transcription there are two additional complexities. First, there are at least 3 RNA polymerases in the nucleus. जो यूकैरियोट्स हैं, तो यूकैरियोट्स में 3 RNA polymerases हैं. There is a clear-cut division of labor The RNA polymerase first transcribes rRNAs 28S, 18S, and 5.8S. The RNA polymerase second transcribes the precursor of mRNA, that is hnRNA, the heterogeneous nuclear RNA, hnRNA. Whereas the RNA polymerase third is responsible for transcription of tRNA, 5S RNA, and SN, small nuclear RNAs. So, second complexity is that the primary transcripts contain both exons and introns. Exons functional part है और introns non-functional part है. So introns are non-functional. Hence they are subjected to process called splicing. अब splicing से आपको non-functional part को remove करना पड़ेगा. Where the introns are removed and exons are joined in a defined order, capping and tailing. और दोनों तरफ, एक तरफ capping, एक तरफ tailing करके हमें उन्हें security भी provide करनी है. HRNA undergoes additional processing called capping and tailing because हमें उन्हें जो अलग अलग एंजाइम्स हैं उनके एक्शन से बचाना है, मेनली एक्सोन्यूक्लियसेस. देखिए यहाँ एक्सॉन और इंट्रॉन दोनों पार्ट है. एक्सॉन और इंट्रॉन दोनों पार्ट होने पर जो आपका इंट्रॉन पोर्शन है उसे हमें रिमूव करना है. तो इंट्रॉन्स को रिमूव कर दिया. दिस प्रोसेस इज कॉल्ड स्प्लाइसिंग. तो स्प्लाइसिंग से आपने क्या किया, नॉन फंक्शनल आरएनए या इंट्रॉन्स को आपने रिमूव कर दिया. इन कैपिंग an unusual nucleotide methyl guanosine triphosphate is added to the 5' end of hnRNA. तो 5' पर आपने क्या किया, methyl guanosine triphosphate को ऐड कर दिया. It is called capping. In tailing, adenylate residues about 200 to 300 are added at 3' end. 3' end पर आपने क्या किया, 200 से 300 आपने क्या किया, adenylate residues को ऐड कर दिया. It is called 3' tailing. Splicing, 5' capping and 3' tailing. इन तीन प्रोसेस के बाद hnRNA mature RNA बन जाता है. It is the fully processed hnRNA, now called mRNA, that is transported out of the nucleus for translation.